ఇంటర్మీడియట్ విద్యారంగంలో అగ్రగామి మన NSR ఇంపల్స్ ఎడ్యుకేషన్ ఇన్స్టిట్యూషన్. ఐఐటి జేఈఈ అయినా నీట్ అయినా తిరుగులేని విద్యా సంస్థ NSR ఇంపల్స్.
Hello students, now let us continue with the concept of respiration. For the last class we have discussed very clearly the differences between aerobic respiration and anaerobic respiration. So for both aerobic and anaerobic respiration, the common pathway is nothing but glycolysis. Now before we start with the aerobic and anaerobic, here what I am trying to tell you here is, for aerobic respiration as well as for anaerobic respiration, what we have discussed, very important point for the last class, the glycolysis is the common pathway which happens in both aerobic as well as anaerobic. So we will discuss glycolysis. For when we start aerobic respiration, we start with glycolysis followed by link reaction, then followed by Krebs cycle, and then lastly we have to discuss about ETS. Okay, so in aerobic respiration, so we have to study about glycolysis, link reaction, Krebs cycle as well as ETS. So when you take aerobic respiration, glycolysis, link reaction, Krebs cycle and ETS, we will start with aerobic respiration. At that time we will start with glycolysis. But before that, let us start with anaerobic respiration. In anaerobic respiration also, glycolysis is a common pathway, and once we complete glycolysis, then immediately what occurs in anaerobic respiration is nothing but fermentation. What occurs in anaerobic respiration is nothing but fermentation. So first step here is glycolysis. Once we study complete about fermentation, then we will start with glycolysis. That means glycolysis is a common pathway for both aerobic and anaerobic respiration. So now glycolysis we will start at the time of aerobic respiration. Presently let us discuss about anaerobic respiration. In anaerobic respiration, first step is glycolysis. What is glycolysis we will come to know at the time of aerobic respiration. Let us begin with fermentation. So before aerobic respiration, let us start our discussion with anaerobic respiration. Okay, let us start our discussion with anaerobic respiration. In that, what is the first step of anaerobic respiration? The first one is glycolysis. Now, what is the second step under this is nothing but what? Fermentation. Now, let us discuss about fermentation. So, glycolysis we will discuss, will start at the time of aerobic respiration. Right? So, we will come to know about glycolysis at the time of aerobic respiration. You know that anaerobic respiration occurs in the absence of oxygen. In that, the first step is glycolysis. So, let us begin with the second step called as fermentation. Let us begin with the next step called as fermentation. Okay, now let us continue with fermentation. Now when you take the fermentation, the process of fermentation, the first time discovered by the scientist Gay-Lussac. Initially, the fermentation process was discovered by the scientist Gay-Lussac is a scientist. Now, he studied the process of fermentation with the help of by using fermentation vessel. Those fermentation vessels are itself are called as Kuhne vessel. Fermentation vessels, what we start using them itself are called as what? Kuhne vessels. Now, so when you take the fermentation is being discovered by Gay-Lussac by using the large vessels in the industries for the extraction of alcohol, and those vessels are called as Kuhne vessels. Later, a fermentation process was demonstrated and studied experimentally, proved by, demonstrated by, with the great effort of the scientist Louis Pacho. Who is the scientist? Louis Pacho. Right. So the fermentation process is being demonstrated by, experimentally studied by the scientist called as Louis Pacho. Now, Pacho observed that, further Pacho has studied, of course, what is called rabies vaccine has been discovered, and preservation of milk by pasteurization method. So till today we get milk packets from the dairy farm, and their milk is preserved by the method called pasteurization that was being invented by Pacho. So till today for the preservation of milk what we use is called pasteurization. Not only that, Louis Pacho also studied in some organisms. So it was identified, there was a discovery of Louis Pacho. He identified in some organisms the shifting of anaerobic condition to aerobic condition. There will be in some organisms the process of shifting of anaerobic condition to aerobic condition itself is referred as, it is observed by Louis Pacho, and itself he referred as, or itself is called as Pacho effect. Itself is called as what? It's called as Pacho effect. What is Pacho effect? The process of transfer of anaerobic respiration into aerobic respiration, or you write down in place of condition, better you write down respiration. Better thing is in place of condition, that means instead of without oxygen, now they started utilizing oxygen. Anaerobic condition you can write, or you can also write respiration. So what is anaerobic respiration? The organisms which are found growing in the absence of oxygen after certain part of their life cycle, after in certain conditions, they shift from anaerobic respiration to aerobic respiration. Sometimes they are anaerobes. When they come in contact with oxygen, they become aerobic. Actually they are anaerobic. Anaerobic means they grow in the absence of oxygen, but then they start growing in the presence of oxygen. Shifting from anaerobic respiration to aerobic respiration observed in some organisms, it was referred by Pacho as, it is Pacho effect. Now, so if you look at fermentation process, now we have two types of fermentation was being observed. Let us talk about types of fermentation. Right. Fermentation, it is of generally it occurs in two types. Let us take the first one is, it is referred as alcohol fermentation. First one it is referred as alcohol fermentation. Let us take another one as lactate fermentation. Let us take other one as a lactate fermentation. So one we call alcohol fermentation and other one we call it as lactate fermentation. Right. So when we take the fermentation, you know that it happens in the absence of oxygen. Now let us take, by the end of glycolysis, what produced here it's called pyruvic acid. So there will be two molecules of pyruvic acid. What is this pyruvic acid? It is nothing but end product of glycolysis. Okay, the end product of glycolysis is nothing but two molecules of pyruvic acid. Now generally pyruvic acid, each pyruvic acid, it contains 3 carbons. So, we will start with glycolysis as a part of aerobic respiration, but try to remember in anaerobic organisms also first step is glycolysis. At the end of glycolysis, pyruvic acid is produced, 2 molecules, each one having 3 carbons. Now, in some organisms it undergo alcohol fermentation. In lactic acid and lactic bacteria it undergoes what is called lactate fermentation. Alcohol fermentation most commonly it is observed in case of yeast. It is observed in case of yeast. What is yeast? Saccharomyces cerevisiae. The scientific name is Saccharomyces cerevisiae. Right. Now we have taken the cultures of the yeast and we want alcohol to be produced, and yeast is added in the culture media, and the culture media is nothing but large vessels having nutrients, means nutrient media. Then yeast is undergoing multiplication process. You know how yeast undergo multiplication process? By budding, right. When yeast is multiplying, suppose if the yeast is increased up to 13%, yeast undergoing multiplication, keep producing alcohol, keep producing alcohol. Suppose if the alcohol percentage is beyond 13%, then it itself becomes poison. If the alcohol percentage is 13%, if it is crossed 13%, then it itself becomes poison. It itself becomes poison. That means when yeast is continuously undergoing the process called multiplication, continuously producing the alcohol, you should not exceed alcohol percent beyond 13%. You should not exceed 13% of alcohol. The alcohol concentration produced, it should be within 13%. What happens if the alcohol concentration goes beyond 13%? Then the yeast, that alcohol percentage itself becomes poison for the yeast cells. All yeast cells will be die, they will destroy, and yeast cells are not there, then alcohol production will stop. So, we have to say that it should not cross beyond 13, we should not reach up to 13%, it should be below 13%. If it is beyond 13%, then it itself, alcohol itself becomes poison for the yeast cells. Yeast cells when they die, then alcohol fermentation will stop. So, this is one precaution we have to take. Alcohol fermentation, it happens in the yeast cells when alcohol percentage is 13%. So, here alcohol fermentation, yeast becomes poison if it crosses 13% of alcohol. That alcohol itself will become poison for the yeast cells. All yeast cells will die, then alcohol fermentation will stop, right. So, now let us talk about alcohol fermentation. Now, in this alcohol fermentation, the first 2 molecules of pyruvic acid, 2 molecules of pyruvic acid plus 2 molecules of NAD plus, right, 2 molecules of pyruvic acid plus 2 molecules of NAD plus. Now, here what we find is nothing but the name of the enzyme is pyruvic decarboxylase, pyruvic decarboxylase. Now, it produces what is called as acetaldehyde. Along with that, there is a release of carbon dioxide. Okay. So, the 2 molecules of pyruvic acid, it reacts with, it will get reduced to NAD plus with the help of enzyme. Enzyme is also very important for all of you to remember. Pyruvic decarboxylase and produces acetaldehyde. Along with that carbon dioxide plus there is a production of NADPH2, very important. So, there is a production of 2 molecules of NADPH2. And further, these 2 molecules of acetaldehyde, what we find here, now the 2 molecules of acetaldehyde, further with the help of an enzyme pyruvic dehydrogenase, with the help of enzyme pyruvic dehydrogenase enzyme, now it produces 2 ethyl alcohol. it produces an enzyme and component called ethyl alcohol. Right. So, what are the things which are produced here? Carbon dioxide is produced, 2 molecules of NADPH2 produced, and finally get synthesized with what is called as the component called as ethyl alcohol. So, the 2 enzymes which plays a vital role in the preparation of alcohol are nothing but pyruvic decarboxylase and pyruvic dehydrogenase. Very important. So, I repeat again, alcohol fermentation mostly we find in Saccharomyces cerevisiae, yeast generally multiply and keep producing alcohol. If it is alcohol concentration, if it is going beyond 13%, alcohol percent itself becomes poison to the yeast cells. So, 2 molecules of pyruvic acid which is produced as an end product of glycolysis. We will start glycolysis now for aerobic respiration. For in anaerobic respiration also first glycolysis occur. So, the 2 molecules of pyruvic acid produced as an end product of glycolysis now undergo, either it may undergo alcohol fermentation or it may undergo lactate fermentation. So, here we have 2 types of fermentations. The first one, alcohol fermentation, when you take 2 molecules of pyruvic acid each having 3 carbons, now it get reduced with 2 NAD plus. Now, with the help of an enzyme pyruvic decarboxylase, it produces 2 acetaldehyde plus carbon dioxide molecule and 2 NADPH. So, here 2 molecules of carbon dioxide. Carbon dioxide is released out, NADPH2 released out, or you can write carbon dioxide release, you can show here, no problem. Instead of writing here, you can show carbon dioxide is coming out. Anywhere you can write, no problem, right. So, 2 molecules of acetaldehyde, 2 carbon dioxide, 2 NADPH2. Acetaldehyde further get reduced with pyruvic dehydrogenase enzyme. Here it produced with 2 molecules of ethyl alcohol. Alcohol is produced, that is why it is called as alcohol fermentation. Next, another one is lactate fermentation. So, when you take the lactate fermentation, so here the 2 molecules of pyruvic acid. So, we say again 2 molecules of pyruvic acid. Now, in this case also we take 2 NAD plus, right, and it get produced with what is called as 2 molecules of lactic acid, and along with that, so you can write 2 molecules of NADPH2. So, what produced here, the end product is nothing but lactic acid. As lactic acid is produced as an end product, that is why it is called as lactate fermentation. So, here pyruvic acid each having 3 carbon get reduced with 2 NAD plus. So, produced with 2 NADPH2, along with that end product produced here, it is called lactic acid, and it operates catalyzed with the enzyme lactate dehydrogenase. What is the name of the enzyme? Lactate dehydrogenase. So, here you find 2 molecules of pyruvic acid plus 2 NAD plus lactate dehydrogenase produced with 2 molecules of lactic acid plus 2 NADPH2. Now, this is called lactate fermentation. Generally, lactate fermentation happens in bacteria, in lab we call, you studied in case of microbes in human welfare. At that time you studied what is called as lactobacillus. What is the bacteria name? Lactobacillus. It is called as what? Lab. Lab means what? Lactic acid bacteria. What is lab? Lactic acid bacteria. It is called lactobacillus. Now, this is generally lactate fermentation. It's, you know that it's nothing but what? It's a type of anaerobic respiration what we study. It's a type of what? Anaerobic respiration. Generally, if you take for example human skeletal system, generally if you take, now at the during exercises, the deep tissue what you find, or in muscles during exercises, in muscles what occurs is nothing but anaerobic respiration, because oxygen is not sufficient. Oxygen is not sufficient, where in deep tissues oxygen was not sufficient and not supplied to the deep tissues. So, in the deep tissue what occurs is called anaerobic respiration. As a result, here it produces lactic acid. Undergo what? Undergo anaerobic respiration. Undergo anaerobic respiration, or we say anaerobic respiration takes place and produces what? Lactic acid. Produces what? Lactic acid. So, here you just have to remember, even in case of human beings, not only in microorganisms like lactic acid bacteria what undergoes lactic fermentation, no. Not only in lactic acid bacteria and some microorganisms, lactate fermentation also happens in the human skeletal system. When this happens in human skeletal system, generally in human beings, in all cells, what occurs is aerobic respiration. But sometimes what happens in the human skeletal system, especially during exercises, when human beings are doing very huge exercises, at that time in muscles, what I wrote here, in muscles, here oxygen is not sufficient to the deep tissues. Oxygen may not be supplied properly to the innermost deep tissues. So, oxygen is not sufficient, not properly supplied to the deep tissues. As a result, within the deep tissues, it undergoes anaerobic respiration and produces what is called lactic acid. So, lactic acid fermentation, even there is a chance occurring in human skeletal system at the time of exercise. During exercises, in muscles, deep tissues may not be supplied with oxygen, then they tend to undergo lactate fermentation as an anaerobic respiration. So, these are the two cases, but here you remember carbon dioxide is released here, but carbon dioxide is not produced in case of lactate fermentation. But what produced here you can notice, that is 2-NADPH2 in both alcohol fermentation as well as in lactate fermentation. Now, let us continue further. So, let us go with the next one, aerobic respiration, right. So, in aerobic respiration, what is a very important first one we have to discuss? Aerobic respiration, we have 4 steps. I wrote very clearly what are they: glycolysis, right, and followed by link reaction, Krebs cycle, electron transport system. We have 4 steps in aerobic respiration. Now, let us start with the first one. In this, let us go with now glycolysis. Let us go with very first one, glycolysis. Now, if you look at glycolysis, is the very first step in aerobic respiration. Glyco meaning here, it's generally glyco meaning it is nothing but glucose, and lysis means breakdown. So, it is a mechanism of breakdown of glucose. What is this? Glyco means glucose, lysis means breakdown. So, we say it is a breakdown of glucose, we call glycolysis. Now, in aerobic respiration, when you take the glycolysis here, now generally we say glycolysis was being studied by many scientists, but 3 people were successful in giving a complete pathway of studying and understanding glycolysis in case of eukaryotes. Generally, you know, the aerobic respiration most commonly, when you talk about respiration, generally anaerobic respiration Anaerobic respiration most commonly happens in case of prokaryotes, right. Coming to aerobic respiration, aerobic respiration occurs in some prokaryotes and in all eukaryotes. So aerobic respiration, it's a characteristic feature of what? Eukaryotes. In all eukaryotes, what occurs is called aerobic respiration along with some prokaryotes. Now let us talk about glycolysis. Glycolysis is being given by the scientist. So we are in aerobic respiration. So here if you take glycolysis is also called as EMP pathway, also called as EMP pathway, or it is also referred as fundamental respiration, or itself is also called as core respiration. Okay, so what are the different terminologies we generally have to remember for glycolysis? Guys, you have to practice glycolysis. Glycolysis is very important, and definitely we have questions which we get in the final NEET paper. So glycolysis has lot of weightage. Very important concept here. What we are discussing is nothing but first step of aerobic respiration. What is that? We are discussing glycolysis. As I told you, glyco means glucose, glucose lysis, breakdown of glucose. So here this is called as fundamental respiration. So for any respiration to start, the first step they need to undergo glycolysis, whether it may be aerobic or maybe anaerobic, but the first step they need to undergo glycolysis. That's why it's called fundamental respiration, or itself is a basis for any respiration, also called as core respiration, or this is also referred as universal biological process, meaning it occurs everywhere in all living organisms. That's why this is also called as universal biological process. Okay, glycolysis occurs universally in all living organisms, the organisms which undergo anaerobic respiration as well as aerobic respiration. That's why glycolysis is also called as universal biological process. Right. Now fundamental, core, universal you understood. Let us come to why it is called as EMP pathway. Why do you think it's called as EMP pathway? Because you have to remember that glycolysis is being discovered by or studied by a coordination of three scientists, Embeden and Meyerhoff and Paranas. Embeden, Meyerhoff and Paranas. Who are the three scientists? Embeden, Meyerhoff and Paranas are the three scientists who studied about glycolysis. That's why glycolysis is also called as what? EMP pathway. Who are the three scientists? Embeden, Meyerhoff, Paranas. That's why it's called EMP pathway. Right. Now let us start with the pathway. So just note down the scientist name. I'll just erase the scientist names. Because I need total board for writing of the cycle. So here you can take the first, it begins with the compound called as glucose. So all of you know it, glucose. It has 6 carbons. And what is the molecular formula we find? C6H12O6. So one molecule of glucose, here it get converted into a compound called as a glucose 6-phosphate. Now a phosphorus is attached at the 6th position. Now it is referred as a glucose 6-phosphate. Right? Initially a one molecule of glucose where phosphorus is attached at the 6th position. So it converted into glucose 6-phosphate. Further, glucose 6-phosphate get converted into fructose 6-phosphate. Fructose 6-phosphate. So here both of them are nothing but isomers. Simply the conversion happens from glucose to fructose. Glucose is one component, fructose is another component. So there is a conversion of glucose 6-phosphate to fructose 6-phosphate. Right? Then here fructose 6-phosphate converts into fructose 1,6-biphosphate, or we say bisphosphate. So here, so they keep converting from one form to other form. Now let us discuss in detail. I repeat again, so take white paper, simultaneously you practice. Glycolysis is very important. One molecule of glucose having 6 carbons here, it get converted into one molecule of glucose 6-phosphate. Further it converts into its own isomer. So glucose and fructose both are isomers. So glucose 6-phosphate converts into fructose 6-phosphate. Now that further get converted with the addition of one more phosphorus. So here phosphorus is attached to the 6th carbon. Now here one more phosphorus is attached, bisphosphate it called. One more phosphorus attached to the first carbon of fructose. Phosphorus is there for 6th carbon. 2 phosphorus are there. So we call fructose 1,6-biphosphate. Right. So even this has 6 carbons. Okay. Now let us discuss clearly here. Now for conversion of where glucose will get the phosphorus which is attached to 6th carbon comes from where. So try to remember, here it utilizes a molecule of ATP, utilizes and gets converted into ADP. So you have to note down very clearly, here utilization is there. A phosphorus is being utilized. Okay, so here phosphorus it's being utilized. One molecule of ATP, it's consumed and there is a formation of ADP. Phosphorus is utilized. So what is the name of the reaction? I am writing name of the reaction this as phosphorylation. What is the name of the reaction? Name of the reaction as phosphorylation. Guys, I hope it's clear. Let me check. Right. So what is that? Name of the reaction is phosphorylation. So ATP, here what happens is ATP, a molecule of ATP utilized. So name of the reaction is phosphorylation. Phosphorus is added at the 6th carbon. So taking this phosphorus, glucose converts into glucose 6-phosphate. Then what is the name of the enzyme? Name of the enzyme is hexokinase. Wherever kinase is there, that takes with the help of magnesium. So for every step of reaction, we are writing 2 things. One is name of the reaction. Second thing is name of the enzyme. Third thing is if there is any energy is utilized or released, we have to mention. So what is the name of this reaction? Name of this reaction is nothing but phosphorylation. What is the name of the enzyme? Hexokinase. Which metal ion is essential for kinase enzyme? Wherever you find kinase enzyme, magnesium is very essential for that. A metal required for kinase enzyme is magnesium. ATP utilized and lead to formation of ADP. So this reaction is called as phosphorylation. Next, so it got the phosphorus. Phosphorus is attached to the 6th carbon. Right. Now, glucose 6-phosphate converts into fructose 6-phosphate. Now, what is the name of this reaction? This name of the reaction is called as isomerization. What is the name of this reaction? Name of this reaction is called isomerization. So in this case, if you look at, so you can take phosphogluco. So here you can write name of the enzyme, phosphorus glucose isomerase. Okay, what is the name of the enzyme? Phosphogluco you can write, or glucose you can write, glucoisomerase. That is nothing but name of the enzyme, phosphoglucoisomerase, because conversion from glucose 6-phosphate to fructose 6-phosphate, it's called as isomerization. Then again here fructose is having only one phosphate that is attached to the 6th carbon. But in the next step we are getting one more phosphorus. From where we are getting one more phosphorus? Again here ATP is utilized, consumed and converts into ADP. So the next step, third step is also called as phosphorylation. Third step is also called as what? Phosphorylation. So fructose 6-phosphate converts into fructose 1,6-biphosphate, and already one phosphorus is there at the 6th carbon. Now ATP is consumed. So here you can take ATP is consumed. Remember, this is also important. One molecule of ATP is utilized, converts into ADP. So phosphorus is added to the first carbon here, thereby it becomes fructose 1,6-biphosphate. So this is also a one molecule of fructose 1,6-biphosphate. So the name of the enzyme is phosphofructokinase. The name of the enzyme is name of the substrate. So here we write phosphofructokinase. Again magnesium come into the picture whenever the enzyme kinase is catalyzing the reaction. I hope 1, 2, 3, 3 steps are clear. I repeat again, glycolysis is very important. I expect all of you practice and try to learn. This becomes very easy. Glucose 6 carbons, now it undergo reaction called phosphorylation, consumes one molecule of ATP, converts into ADP. Now it takes phosphorus attached to the 6th carbon, it converts glucose 6-phosphate with the help of an enzyme hexokinase. Now this glucose 6-phosphate now further converts into fructose 6-phosphate. It undergoes isomerization with the help of an enzyme phosphoglucoisomerase. Next, fructose 6-phosphate, by consuming one more molecule of ATP, it becomes fructose 1,6-biphosphate with the help of enzyme phosphofructokinase, and the name of the reaction is phosphorylation. Now let us continue further. Now here it undergo next step. In the next step it get broken here the compound. So the name of the reaction we write cleavage. It undergoes cleavage. As a result of cleavage here it produces two compounds. What are the two compounds it produces? One is nothing but DHAP having 3 carbons, right, 2 molecules, and other compound is nothing but 2G3P having 3 carbons. Okay, this is also triose. So how many carbons? 6 carbons splits into 3 carbon triose compound, 3 carbon triose sugar here, right. So what is this DHAP? Did you remember? We studied in photosynthesis, right. So what is this DHAP? Dihydroxyacetone phosphate. What is the dihydroxyacetone phosphate? DHAP we call dihydroxyacetone phosphate. How many carbons? 3 carbons. How many molecules? 2 molecules. What is the other compound produced here? The other compound produced here G3P having 3 carbons. How many molecules? 2 molecules. What is G3P? You all know it very well. It is called glyceraldehyde 3-phosphate. What is the name of the compound? Glyceraldehyde 3-phosphate. Okay. The fructose 1,6-biphosphate 6 carbons, it gets split into 3 carbon DHAP and 3 carbon G3P. Now what happens here? These both are isomers, right? Now further it becomes reversible. Now with the help of an enzyme, triose phosphate isomerase, with the help of enzyme triose phosphate isomerase enzyme, they undergo the reaction called as isomerization, meaning DHAP can convert into G3P, and further it participates in the process called as glycolysis. Further it participates in the process called as what? Glycolysis. So if you remember up to here, the 6 carbon compound, it get broken down into DHAP and G3P, right? So this is an important step. Fructose 1,6 biphosphate having how many carbons? 6 carbons, broken down in 3 carbon component, 2 molecules of DHAP, dihydroxyacetone phosphate, and another 3 carbon molecules centers into another compound called G3P, glyceroldehyde 3 phosphate. Very important, and this is reversible reaction. DHAP further converts into G3P with the help of an enzyme. Enzyme is very important, triose phosphate isomerase, and the name of the reaction is nothing but what? Isomerization. Right, up to here I hope all steps are clear. Just first, if you are feel difficult, first you write down blue colored boxes, what conversion after what, how conversions are taking place. In every step, note down the enzyme and name of the reaction. See the places where ATP utilized. In the first step, ATP is consumed, leave one step. In the next step again ATP is consumed. Next breaking, breaking itself is called as what? Cleavage. So DHAP converts into G3P. So this is a reversible reaction. Now let us continue further.